Jeroen Krijgsveld, German Cancer Research Center. Enhanced proteomics workflows with paramagnetic beads for ultrasensitive proteomics.
Hello everyone. I'll be taking you into the world of proteomics and explain to you how we are trying to get this to the next level. So proteomics is the part of science that tries to identify these thousands of proteins in cells and tissue and try to understand how these regulate cellular processes and how this is disturbed in disease. Now really the core technology here is mass spectrometry, which has propelled the entire field, but we are still facing an important bottleneck, and that is in sample preparation. So this is the part where we need to extract somehow proteins from these tissues in a way that we can introduce them in a mass spectrometer. And this is done traditionally in a multi-step process where proteins are extracted, digested, and cleaned, and then peptides are filtered and then sprayed into a mass spectrometer. So this is, in a way, very inefficient, and it suffers from a number of disadvantages. So we have revised this process by radically changing this process of sample preparation, making use of magnetic beads, where we can combine all these processing steps in a single vial, where we mainly benefit from increasing speed and sensitivity, which I'll show you in the next few slides. So first of all, so this is an unbiased method. So it's not targeting a particular protein. It aims to purify all proteins in a sample. This you see on the left side here, we really get everything, from the very big proteins to the very small ones, so there's no selective bias. In the middle image, you see that we can do this in a wide variety of reagents and detergents. So this gives us very great flexibility in how we extract proteins, for instance, in a very high concentration of SDS, and remove it afterwards, making use of these magnetic beads. Third, on the right side, you see we can really benefit from the magnetic properties of these beads. Right? So we can now implement this whole methodology on a robotic platform and using a technology that's actually been used for a very long time in genomics, but that is very new to proteomics now. But very importantly, we gain in sensitivity. Because we operate in a single vial, we don't suffer from sample losses. Shown here that we can, from a very small amount of material, we can identify a large number of proteins, and we can even quantify them. So, in summary, what we have is a robust workflow that is universal, that can be applied to a wide range of proteomic applications. It is scalable from very low amount to large amounts. It is fast, and the throughput can be very high if we implement this on a robotic platform. So this meets actually all the demands that we are currently facing in clinical proteomics, and no surprise, this is exactly the direction we'll be taking in the future. Thank you very much.